Very pleased to have this. Oh, all right. Thanks, Irina. So very pleased to have this opportunity to introduce to you Chrystalina Antoniades. I'm happy to have known her since she was a graduate student with Roger Carpenter at Cambridge University. And for those who may not know, Roger wrote the book on eye movements, literally. He was a delightful colleague and friend, and we miss him. After earning her PhD in 2009, working on biomarkers for Parkinson's and Huntington's disease, she joined the well-known clinical neurology group at Oxford run by Chris Kennard. Today, she's the Associate Professor of Neuroscience in the Nuffield Department of Clinical Neurosciences, where she runs the Neurometrology Lab. You students can look that up. It's a good word. So as PI of a £2.9 million grant from Merck, Sharp and Dome, supporting the Oxford Quantification and Parkinsonism study, her research team is developing novel ways of accurately measuring behavior in patients with neurological disorders. You know, the basic goal is to develop measures of brain function that are just as efficient as many measurements today we have for heart or lung function. And so by developing these new approaches to measure neurophysiological biomarkers, we can replace these old subjective clinical rating scales. Beyond her scientific leadership, Chrystalina has been deeply involved in academic leadership. She's a fellow and a dean of the new Rubin College in Oxford University, which is taking a lot of her time. And yet, in more of her spare time, Chrystalina has launched an art and neuroscience project at the Ashmolean Museum at Oxford. For this, she received the inaugural Vice Chancellor's Award for Public Engagement with Research. So it's a special treat to have her with us today. The students are really going to enjoy conversing with her later. Welcome, Chrystalina.
Thank you, Jeff. Thank you for the kind introduction. It's a great pleasure to be here and to meet everybody and to share a little bit of our work. And I hope at some point I'll be able to visit your institution. I can see a lot of interesting work, and Jeff, with you there, I'm sure it will fly up very high. So I'm really looking forward to seeing what the next few years hold ahead. So as Jeff has very kindly introduced, I'm a clinical neuroscientist, and my area of expertise is the development of biomarkers in neurodegenerative disorders. My specialist interest in Parkinsonism and Parkinson's-alike disorders. Today, I'd like to tell you a little bit about our work on quantifying Parkinson's disease using eye movement, so oculomotor function, but also tell you a little bit about our work on digital phenotyping using digital technology coupled with the eye movement recordings. And as Jeff has mentioned, I grew this on loving eye movements really from one of my supervisors when I was in Cambridge, the late Professor Carpenter, who was the oculomotor professor of the whole Cambridge, and got to introduce me to how wonderful it is to look at eye movements, how much we know physiologically about eye movements, but also how to use your intuition and your own expertise to use eye movements and anything else you want to look at when you're doing your graduate degree, which I would encourage all the graduate students in the audience to consider with their respective areas. And just to set the scene, in particular for those of you and the students that might not be familiar with this neurodegenerative condition, Parkinson's, along with Alzheimer's, as we know, are the commonest neurodegenerative disorders, with the number of the aging population increasing. PD patients present more often, and in some instances at younger ages. And Parkinson's is classically defined by a pathology which targets, I hope you can see my cursor here, the dopaminergic cells in the substantia nigra, and these cells acquire alpha-synuclein Lewy body pathology, which you can see in the figure here. And whilst this is a very useful approximation, if you like, of Parkinson's itself, we know that if we give dopamine drugs, for instance, which target that particular network, some patients may do very well and have a decent quality of life, but we know that this is not really the whole story. And in fact, Parkinson's was firstly described by a British physician, James Parkinson, back in 1817. He didn't call it Parkinson's at that time. He called it shaking palsy. And here in the Bodleian Library, which is the biggest library in Europe, which we're very lucky to have here in Oxford, he wrote it in this, what he called the essay of the shaking palsy. And this is actually an actual copy of his work, which I was delighted to be able to have him read, because you can feel what he was sort of writing when you can see the old sort of a little essay that he wrote. And in fact, we haven't really moved miles ahead until recently. So in 1886, another a neurologist, researcher, and artist, Sir William Richard Gowers, drew this fantastic artistic illustration of what he believed a Parkinson's patient looked like. So you see an older person, a frail person, a person who's sort of leaning at the front. You can see this is a disease of the older, which we know now it's not. And he's having this kurtosis. He's sort of, as you see, that he's got bradykinesia, which is the Greek word for very slow movement. And it's everything we thought all these years that Parkinson's, that's how everybody who has Parkinson's should look like. But in fact, it wasn't until sort of the 19th century that another neurologist, Jean-Martin Charcot, that began calling it Parkinson's disease. And it took about 100 years for people to realize, well, researchers and physicians, I guess, to realize patients were losing brain cells. And in the 1960s, it was discovered that the levels of dopamine were much lower in Parkinson's patients than in healthy controls. And I'm delighted to see that essentially in the recent times, and this is a wonderful schematic by one of the emergency physicians that I've had the pleasure of working with on the art and neurosciences, that I'll tell you at the end, who is a person who has Parkinson's and is what we call early onset, because he was diagnosed in his early 40s. Basically recreated, if you like, what Gowers had originally put as Parkinson's, which is describing both the motor part, so things like tremor, bradykinesia, rigidity, and so on, that we know and physiologically people have described over the years, but also the non-motor, things like apathy, depression. I was talking with one of your colleagues about all of these, and all the things that we know are there, but we haven't really looked into them, including sleep on one of the legs here, that are of extreme importance, especially if you're looking at the sort of pre-symptomatic period for Parkinson's. There is no cure for Parkinson's, but there are treatments that help improve some of the symptoms, and thus the quality of life of the patients. On the one hand, we have various medication treatments, the most popular one including levodopa, that can have an effect with a therapeutic window, I guess, time frame of 5 to 10 years. Some of us agree, some of us disagree. While on the other hand, we have a surgical technique, which I believe some of my colleagues there might know, the deep brain stimulation, which involves electrical stimulation of deep brain areas that can relieve symptoms. Just very quickly, on the left-hand side, here is a stereotactical frame that the neurosurgical colleagues use for the accurate sort of placement of these electrodes. Here is a scan, and I don't know if you can see the two little dots, the white dots, those are the tips of the electrodes. In this case is a patient that had subthalamic nucleus, so STN DBS. And on the right-hand side, you can see what these electrodes, and I guess for the graduate students in the audience, just to see what these electrodes actually look like. So they're placed under on the skin, on the skull, and on the bottom you can see the tips of these two electrodes. That is what you see here, sort of shown as white matter. And essentially what you connect this to is to a small battery, which looks perhaps half of the size of my iPhone here, which is placed under the chest, something like what we call brain pacemaker, which is providing electrical stimulation. And students always email me after talks like this asking me, can I see what a patient looks like before and after? Let me just show you. This is courtesy of the BBC and Paul Mayhew Archer, who is one of the scriptwriters, a person who has Parkinson's and has done a documentary with our colleagues here to show what it looks like when the stimulator is switched on and off.
John Radcliffe Hospital in Oxford, where the deep brain stimulating device thing
I hope you can hear
was
the
put
sound.
in. You're going to switch it off?
Yep.
Certainly can, yeah.
Okay, over to you, John.
So these days, this is done wirelessly. This is a lovely gentleman who came in with a tremor that you can see here. He had a very successful STN DBS. Again, wirelessly.
We'll put it back on.
Stimulation will be switched on.
And you can see
There you go. And you see
almost straight
when
away.
it works really well.
That is absolutely astonishing. It's fantastic.
And I guess the take home message just to get you through a little bit of the deep brain stimulation targets here. There are various areas that can be used for deep brain stimulation for Parkinson's, essential tremor, and other movement disorders. I'm listing here the ones that we know a little bit more on their physiology, I guess, and they're more related, if you like, to some of the eye movement work and some of the gait work that we've been looking. For instance, you can see the thalamus, the pendiculopontine nucleus, or the PPN, which a lot of the older work refers to, especially on macaques as well, and problems with gait and walking. And the mostly used ones, the globus pallidus internus, the GPI, but also the subthalamic nucleus, which is the nucleus that most colleagues around the world are most familiar and have used in terms of DBS. So some of the earlier work we have been doing has been based on trying to quantify eye movements in this patient population I've been telling you, and compare it with individuals who don't have a neurological disorder. And the rationale for this is that eye movements, it's sort of inherently attached to the basal ganglia areas that are involved in saccadic generation. And we know, I'm just giving you a very quick glimpse here, that the STN is sending excitatory input into the substantia nigra pars reticulata. And of course, the superior colliculus is receiving tonic inhibition from the substantia nigra pars reticulata, and in turn is inhibited by the caudate nucleus. And Parkinson's, being a neurodegenerative disorder of the basal ganglia, obviously is very, very closely related with, if you put it, yes, if you have a drug that is acting on this network. So of course it makes sense to start looking at eye movements, which we and others have been doing for many years. A few years back, when I was doing my PhD at Cambridge, a group of us from people who are looking to move from bench to clinics really developed and validated a standardized testing battery among laboratories for one of these oculomotor tasks that I'm sure some of you are familiar with, the antisaccadic task. Essentially what we did here, we wanted to use something that we could take out of our labs and discuss with all our clinical colleagues whether this could be used and take it into clinic, because essentially what I'm talking about here is a translation between what you do in the lab, what you could be doing with your macaques, what you could be doing down with cell biology, molecular biology, and so on, looking at sort of neuronal activity to the point that you want to actually test the patient, and the patient can actually perform the tasks that we are sort of asking them to do. And what we did is that we proposed this international standardized antisaccadic protocol, where we suggested that you start with a block of prosaccas. In this instance, we said, let's start with a 60 trials, followed by 3 rounds of 3 blocks rather of 40 of antisaccades, and then followed by 60 prosaccades. And this was essentially our effort to bring some standardization into the madness of all the different protocols that we have. It's by no means comprehensive, by no means it does what it says on the box, but at least it provides us a little bit of a starting point for the colleagues who might be a little bit worried about entering into the world of oculomotor testing, especially I guess the clinician colleagues who might not want to sort of get into more detail on the oculomotor function. And what we studied, just a couple of studies just to show you how we've used this oculomotor paradigm. So here we used a simple antisaccadic task. For those of you in the audience that might not be familiar with the antisaccadic task, although I gather most of you are, is looking away from a visual stimulus, which you have to inhibit a first movement and then sort of voluntarily start another one to look away from something that you're presented. For instance, if I ask you to look away from your screen or from wherever you are joining us today. And what we did for this task is that we looked at a group of Parkinsonian patients who had deep brain stimulation with electrodes in 2 different parts of the basal ganglia. I've shown you in an earlier slide, the subthalamic nucleus and the globus pallidus internal. I'll be referring them as STN and GPI. So dopaminergic medication alleviates many symptoms, but many patients have more complex problems, such as difficulty with initiation of movement, inhibiting unwanted movement, decision-making, et cetera, et cetera. And these are not really improved by medication to the same degree as the simple movements we see. And in order to understand why this happens, we must first consider, if you like, the way that the motor circuits in the brain are organized. There are several circuits that deal with different aspects of movement. For example, if this red circuit here, hope you can see my cursor, we call it the motor or the oculomotor loop. It deals with simple movements that are often reflexive and very often subconscious. The blue one here, the prefrontal lobe, we call it, is necessary for more complex things such as inhibition, initiation, working memory. There's a huge list that can go in this side of the story here. And in order for more complex aspects of movement to be realized, information must flow from this loop into this loop. And this happens in the basal ganglia, as we all know, in the striatum, that it's the area that is most affected by loss of dopamine. And of course, lack of dopamine causes nerve cells in the striatum to fire randomly, which can disrupt the information flow. And what we found, very briefly, on the top row here are the healthy controls with pro-saccades on the left and anti-saccades on the right. To our surprise, we discovered that the performance was quite different between the groups. Specifically, patients with GPI stimulation show a marked improvement in performance. And I'd like to draw your attention to the GPI graphs. This is from one patient who had GPI off and GPI on, and this is from another patient who had STN-DBS off, and STN-DBS on. And just to show you the number of sort of different number of errors when the stimulation was switched on. So everything under 0 here are errors, anything above are correct responses. Look at how many errors there are when the patient at GPI DBS with the stimulator switched off. You switch the stimulator on, you leave it for a little while, look at how few errors they have, which this has led us to propose a new model of how DBS of the globus pallidus is working. And we're currently investigating this further, and we've been following these patients out for the last few years. And I guess my last slide on this is very much about striatal noise, which I'm sure my neurophysiology colleagues here know a lot more than we do, because there's so much you can do in the human brain and in the operating theater, really. So noise obviously prevents information flow from higher circuits, and it can really disrupt the signals, I guess, that one could be getting, especially during an awake deep brain stimulation surgery. And one of the sort of areas that we've been focusing is what is the role of this information disruption, if you like, with freezing of gait, with speech problems, which you know that these patients have, but also things on the cognitive side with mild cognitive impairment and other cognitive areas that we should look at when we're looking at Parkinson's. I'll leave that there, happy to answer any questions for that part of the study. I'll fast forward in the last year, and a recent study we've published on the effect of levodopa on saccades, and this is part of a study I ran here, the Oxford Quantification in Parkinson. I'll come back to it in a couple of slides. What we wanted to look at for this particular study is to characterize the effects of anti-Parkinsonian medication on pro-saccadic and anti-saccadic parameters in this large cohort of Parkinson's patients and age-matched controls. So we looked at 48 patients and 44 controls. By controls, I mean people who don't have a neurological disorder. And in most cases in such studies, they tend to be the spouses of the patients themselves, so the husband or the wife. And if they don't have a spouse, it's the partner or someone, a friend who comes along the clinic to see them. And we looked at latencies, amplitudes, velocities, and directional errors. And what we found here was that the pro-saccadic latency is significantly prolonged by dopaminergic medication in Parkinson's. And this effect size is comparable to the difference between Parkinson's medication off and healthy control values, which is what you see here. There was no statistically significant change in any other saccadic parameter. So if you look at amplitude, if you look at peak saccadic velocity, all of those seem to be absolute same with on and off medication and medication, disease and healthy controls. But of particular note, antipsychotic latency was almost exactly the same on and off medication, while being almost 20% longer in Parkinson's patients compared to the healthy controls. And if I zoom in a little bit more here, what you see is that these are not affected by having Parkinson's. These are affected, by having Parkinson's, but not by the medication itself, which you see here between the on and off medication. This potentially makes them an excellent biomarker for trials of drugs that aim to slow down disease progression. And if I move you to the other side of the antipsychotics, you can see the no change with medication between the on and off of medication. But you can see the effect of disease if you compare people who are on their normal medication, what we call them as on, but this time medication, and the healthy controls. So just to summarize, prosaccadic latency is significantly affected by dopaminergic medication, which may complicate its use if we're looking to use it, which we have been, as a biomarker in a drug trial. On the contrary, antipsychotic latency has proven to be of particular interest in this regard because it shows a large effect with no medication effect. And we went a step further because one of the reviewers at this journal wanted us to look at the meta-analysis of what has been done to date. And in fact, was a very fair comment, and I think it's made the paper much stronger in a way, because we looked at the effects of antiparkinsonian medication on prosaccadic latency. So panel A are the effects of levodopa treatment only, and panel B down here are the effects of combined antiparkinson medication. And in both cases, and you can look at the OxQuip study and also see how few studies there are since 1989 that have done an on and off medication, and to show how difficult it is to do studies like this. Ethically, you're not allowed to ask your patients to come off medication. And obviously you had to apply for a special ethical approval to ask the patient, stop taking your medication for a day or a few hours before you come and see us. And then we put them on medication once we tested their eye movements. And in both cases, both with only levodopa or a combination of different antiparkinsonian medication, there is prolongation of latency, as you can see, to a greater degree when you've got multiple medications rather than levodopa alone. And this was really interesting because, again, looking at what other colleagues have done with different populations and with slightly different protocols, if you like, but along the same line, it's very interesting to see where our study was falling with this larger number of people. And it was nice to see that we were within the range, if you like, of what other colleagues have found on this. And again, it's been very interesting because we've been asked by various companies that are developing drugs for Parkinson's and Parkinson's, like whether eye movements then could potentially be used as a biomarker and what the value is in having eye movements, which is very well quantifiable in terms of the technology that we have these days as a potential surrogate market as well in drug trials. And we're working with a couple of companies with us, which is fantastic to see the work moving from bench to clinical trials at the moment. I'm going to shift gear a little bit here and just show you what we've been doing in this study, which does include movements, but also some of the other digital technology. And then I'll bring it all together really to show you how using eye movements plus other digital technology can be tremendous when you're trying to develop biomarkers. So the last study I've shown you, the last paper, was very much part of this OxQuip study, which is a short for Oxford Study of Quantification in Parkinsonism. There have been many biomarker studies in the area of neurodegenerative disorders, and OxQuip is a longitudinal study that follows patients with both Parkinson's, but something also called progressive supranuclear palsy, which is an atypical Parkinson's, we call it in neurology, and it's in fact a tauopathy. So a very different type of disorder. It's very close pathologically to Alzheimer's and dementia, as opposed to Parkinson's and a basal ganglia disorder. But it does mimic the disorder. It does mimic Parkinson's, which, sorry, I've lost my cursor, bear with me. Okay, I found it. Sorry about that. But it does mimic the disorder to a degree that a lot of patients are misdiagnosed to begin with for a few years. The trouble is that PSP is a relatively quick neurodegenerative disorder in the progression. So patients do have 5 to 6 years, in better situations up to 7 or 8, where they decline quickly, leading death. And obviously it's very important to be able to give accurate diagnosis to our patients, and also for drug trials, how we screen patients to go into different, taking into different drugs. What we're doing with this trial is that it's an observational trial. We don't have any drugs in these, and we are following patients very frequently with assessments every 3 months, with the aim of identifying a signal of progression at the much shorter time scale in order to be able to rapidly screen newly developed drugs. This is important because only a small minority of drugs that have already been developed actually make it successfully through trials to the market, and developing and trialing, if you like, such drugs consumes a vast amount of resources. So the aim is to kill off research and divert both time and money to the next target. And in OxQuip, we're measuring neurophysiological and cognitive signals to find physiological correlates over a time scale of 3 to 6 months. So we're looking for a very early signal, if you like, to be able to either kill the marker or continue it into the next line of development. Of course, we're also focused on ensuring that the baseline diagnosis is as accurate as possible, because it obviously is very critical to get the right patients, if you like, into the trial to begin with. And there are 3 components, to my mind at least, to better disease measurements: more sensitive tests that are objective, not observer dependent, and tests that are quantifiable in linear scales rather than ordinal scales to permit more sophisticated and accurate statistical analysis. And of course, technological advances have enabled us to measure some neurophysiological signals very easily from bench to clinic. In OxQuip, we measure conjugate, rapid conjugate eye movements. We use portable devices. We use devices that you see here. We can get hundreds of trials measured within a few minutes, and of course, patients can to do. Similarly, we are using devices, as you see down here, rating scales. What we the UPDRS, this is short for Unified Parkinson's Disease Rating Scale, using an accelerometer that is placed firstly on the finger, as you see here, and then on the side of the shoe. And what I mean by clinical rating scale, I mean questionnaires, that if you come to the clinic, we'll ask you to do movements like this: touch your nose, touch my finger, get you out from the chair, walk to the end of the room. And we have to use our judgment and our training to tell you whether it's a 0, which is normal, or 4 or 5, which is abnormal, and so on. And this is really not good enough, especially in 2022, where we could be doing this much more accurately. We also look at cognition. This is a cognitive tablet, but unfortunately my video is not working, but it's essentially moving from pen to paper into getting reaction times, into getting, coupled to eye tracking and so on, which we eventually hope that these computerized tablets will replace the classical and very inaccurate pen and paper tests that we're used to doing. And last, but certainly not least, we measure gait using a body-worn array of inertial sensors, so IMUs, containing both accelerometers, gyroscopes, and magnetometers, to be able to measure our patients accurately. And I'll show you a study we've done in just a moment. And just very briefly, to show you what it looks like, I hope this works. Yes, my videos work. This is how you can use portable oculometers in the clinic. This is part of the neuroscience, our patients' clinics here at the John Radcliffe Hospital within the University of Oxford hospitals. And I can go to my clinic and I can take this, so I don't really need to take my patient down to the lab on an island, for instance, which we do if we want to do something more complicated, because these are sort of the less complicated tasks. But I can use the wall, as you see here, and carry out a very quick pro-saccadic task, anti-saccadic task. I use the room that is dimly lit, so I can keep my recordings as accurately as possible. But you can see the patient can tolerate this really well. This is also what the sensors look like when they're placed on the wrist. So another one of our wonderful patients, who gives permission to show pictures like this, is wearing this sensor here. We also have one on the lumbar, sort of lower back, one on the chest at the front. I'll show you them in a moment, all of them, and one on each feet. So we can have a sort of a portable way of looking at their gait. What you see here is the wealth of data. We're in the world of big data now, which is good and bad, because with big data, you need to know how to interpret it very carefully. This is a 3-axis accelerometer, 3-axis gyroscope, and 3-axis magnetometer on each sensor. You can imagine the wealth of data that you get from a 2-minute walk. And we've been working with our bioengineering friends here in Oxford to be able to understand how we can do the famous machine learning that it took me a while to understand how you get your head around this. So let me share with you some of our results on cognition and digital technology now very quickly. For gait, that supplement very nicely eye movement work we've been working on, and how we are now coupling eye movement with both gait and cognition, and how it's important to think outside of the box when you're trying to do translational research. This paper that's been recently accepted in the BMJ Open Neurology. This is very briefly the, very happy to send this to anybody who's interested to read further. This is very much looking at the smaller population of these atypical Parkinsonian patients. We had 28 PSP patients, so these patients that in fact have a tauopathy. And what I want to draw your attention is down here to this graph H, on their oculomotor function. So even clinically, and this is a combination of looking at their eyes horizontally, looking at their eyes vertically, and sort of summing up the two, even at visit 4, which is about 9 months from the disease, and every visit, so visit 1 is baseline, visit 2 is 3 months, visit 3 is 6 months, and so on, 3 months thereafter, you can see that there is a little star there indicating statistical significance. That's at the level of 0.05 and 0.008 for the 2 stars here. So even on a clinical level, you can see that their eye movements are proving to be an early signal. And what we've been trying to do is quantify this early signal using oculomotor technology, oculomotor testing rather, with the technology that we're given. But it's very interesting because even from very simple ways of saying, follow my pen, look up to the right, look to the left, initiate and elicit a voluntary eye movement, and the same with the vertical component, can give you tremendous information. Which obviously with digital technology and the portable oculometers I've shown you, it's even more accurate, and it can really be used as a marker of progression. And the last one I want to show you here is panel, the graph J, which is showing clinically again, if you look at an item with this, within this clinical rating scale, we call it the PSPRS, is again, it's a questionnaire we use neurology to assess people who have this typical Parkinson's, and what we call here the gait midline, even earlier at visit 3. So this is our 6 months, we're getting significant changes. And what we've been trying to do is see, can we then replicate this using digital technologies such as the sensors that I've shown you, both from an eye tracking perspective, but also from a gait perspective. So just a couple of slides on the cognition here and how we are coupling now these with oculometers, so we can see what they're choosing when they're looking at this. I've told you that PSP may cause rapid cognitive decline, and we have no clinical tests that may be able to pick this up. Digital testing yields new insights, and therefore using this digital testing hopefully will help us sort of separate which ones are real PSP patients and which ones are real Parkinsonian patients. And what we found in what we call this cancellation task, which traditionally we've done it with pen and paper, is the analysis of the path taken by a patient completing this task is very revealing, whether the patient was able to follow a very clear strategy or not, as you can see from these tasks, two parts here. And essentially we name them as adherent if they were able to follow what we asked them to do, or non-adherent if they couldn't keep to a clear strategy. We're currently coupling this with oculomotor testing. So we're putting in an oculometer and we're trying to make it work to see that the output from our eye movement recordings actually tallies with the cognitive testing, so we can match their reaction times and their behavior, if you like, with the cognitive task. And just very briefly, what we did in this paper that was accepted in Movement Disorders Clinical Practice, we described a novel cognitive measure that we called the strategy adherence. And it was derived from this analysis of how a subject completes this well-known cancellation task. We use something called the OCS Plus. It's a fantastic tool that was developed by my colleagues in experimental psychology, and is a tablet-based cognitive testing tool. And just to emphasize on this was not developed for Parkinson's. This was developed for stroke patients. And when I met one of the late sort of heads of experimental psychology a few years ago, we thought, let's just see if we can use this in Parkinson's patients. And you know, it's done wonders in terms of understanding some of the problems you have with cognition. So the participant is presented with a screen displaying 30 vegetables, 10 cabbages, 10 peppers, and 10 carrots, mixed with 30 fruits, 10 bananas, I think 10 pears, and 10 apples, if I remember correctly, and has to select the fruits with a stylus. And plotting the selection order reveals the system used by the participant to work their way through the task. And some proceed in an orderly geographic pattern through the screen of symbols, other cancel by category. I've seen patients canceling apples and canceling pears, but participants follow any such strategy throughout the task are deemed strategy adherent. Those switching strategy or to a random cancellation partway through or cancelling randomly from the outset are deemed as non-adherent. If I go back here, these are the non-adherent ones and these are the adherent ones. And what I'm really excited of what we're currently doing is we're trying to see where are they looking if they're adherent and where are they looking if they're non-adherent, because I think that will give us a better understanding of their oculomotor function, if indeed there is a cognitive decline, and whether there is any correlation. There might not be correlation in some of these cases, and we're not expecting all of this to correlate, but it would be nice to see where they're looking when they're performing this. So we studied these 27 PSP patients, the ones that look like Parkinson's, but they're not. We looked at different clinical phenotypes. We looked at also a bunch of, I think there were 51 idiopathic Parkinsonian patients and 20 or 21 healthy controls. Participants were tested 5 times at 3 monthly intervals, as I've shown you before. And what you see here in these 2 graphs is what happens after baseline. So if you look at baseline here, 52% of the PSP participants were strategy adherent versus 96% of the Parkinsonian patients and 100% of the control, so patients who didn't have any neurological disorder. The strategy adherence rate for PSP decreased rapidly, as you see this drop from 52% at visit 1 to 30% to 22%, 15 to 11 from visit 2 to visit 5. And the deterioration from baseline in the PSP group was significant at all visits. And once an individual lost strategy they never regained it in any subsequent visit, which was an interesting finding for us, suggesting that perhaps there is an irreversible crossing of significant threshold in cognitive decline. That's one of our hypotheses. We might be wrong, and we're trying to prove this again, coupling into oculomotor one, but this is what our results really sort of are indicating at the moment. There was no significant difference between phenotypes and strategy adherence at any other visit, and we use the Fisher exact test for this to sort of understand this. And down here you can see sort of the conventional measures of what we use, MOCA or MMSE, which is the mini mental state examination that you would use, that they're all sort of pen and paper. And this is an extremely strong signal compared to what I've seen in terms of cognition. And it's an extremely strong signal of progression, and a potential disease-modifying drug could be shown to perhaps successfully retard the progression of the cognitive effect in PSP in a short period of time. I'll leave this here because I want to share with you this last study on sort of the gait technology and then bring it all back together. So we've looked at eye movements. We've looked at a little bit on the cognitive tablet and how significant results can look like if we're really using tablets. And of course, we're exploring the combination between eyes and cognition. And lastly, I want to show you a little bit on how we can accurately sort of discriminate, if you like, between someone that looks like they've got idiopathic Parkinson's disease and someone that doesn't and has PSP using wearable technology and machine learning. Again, we've worked, this is a paper published in Gait and Posture, this, I think, 2020. And we've worked very closely with our bioengineering friends, which we're still working with Professor Tarasenko, who have expertise in machine learning, deep learning and AI, who without this collaboration, actually, we wouldn't have been able to do this discrimination between the two disorders. So we investigated a wearable sensor array coupled with machine learning approaches as a means of disease classification. We analyzed gait in a group of people with PSP using a body area network of wearable accelerometers, and a bit more detail on the exact, if you like, methods that we used here. All participants completed a 2-minute walk on the same straight line and level surface to record their gait. Next, we measured sway, and participants were asked to stand upright and as still as possible for 30 seconds with their eyes closed on a firm surface. And we put a template on the floor to ensure that all participants' feet were the same distance apart. And then we had a timed up go that was repeated 3 times. So if I move on to the next slide, it might be easier to visualize them. This 2-minute walk is in a larger corridor. We just do things for recording purposes and to protect patients' identities here. This is Lily, my wonderful PhD student here, who's been doing a lot of this work, both on gait and eye movements. She's wearing all 6 sensors, 1 on each arm, 1 on each foot, 1 on the front of the chest, and 1 at the lumbar at the lower back, where we see what they're doing in a 2-minute walk. This is the sway, where she stands with this separator. So we have the exact separation for each individual, because we know how how further apart we move our legs could affect how we are swaying in our position in terms of walking. And then this down here is a timed up and go, what we call the tag, where the participant is to get up from the chair, do 180 degrees turn and sit down on the chair. And again, a very exciting part we're looking at here is coupling an oculomotor that we're putting on the patient along with these gait sensors to see what's happening. So what we identified here very briefly, we looked at 17 distinct gait parameters that separate PSP from PD. And all I want you to take away from this slide is looking at how the healthy controls and Parkinson's patients are down here, and look at the PSP patients. We couldn't do just looking at their gait, just walking up and down in our corridors here at the hospital. We managed to, improve diagnosis in a way with an accuracy of 90%, and this is better than any other test available at the moment, including things we've done in the past with imaging, with blood, with CSF results, and so on. And just look at the accuracy, sensitivity, and specificity up to 90%, and accuracy 88% when you use all 6 sensors on the feet, on the arms, on the chest, and then on the lumbar. And just to give you a little bit of information of what it looks like, an example of some of the data available from the gait and sway parameters, you can see the parameters derived from the tasks overlaid onto this normal range here, shown in gray, which make the features that are abnormal very, very clear. So all of this is for the lower limbs, so left and right legs, and this is left and right arm. If they're not within the normal range of this gray, it means that they're exhibiting some sort of abnormality. And of course, the sway. So this is a sagittal versus a coronal axis here. And anything within the ellipse, we consider that you expect this sway. But again, you can see the zigzag lines are indicative of someone really swaying when we're asking them to close their eyes for 30 seconds and just stand still, which for Parkinsonian patients is proving extremely interesting if we're looking at correlating some of these results. And of course, here you can see the parameters derived from the timed up and go task. The parameters from all the tasks were combined in our analysis, but with accelerometers and magnetometers and gyroscopes embedded in these IMUs, we can see exactly duration of turns, exactly how long it takes an individual to stand to see, stand to sit and sit to stand and so on. And again, I've shown you sort of the raw data that we get out of all of this, and we are doing a lot of machine learning with our colleagues to be able to understand some of the raw signals that are coming out. And this is my last slide on this, where we show that the key parameters emerging from an analysis discriminating PSP, Parkinson's patients and healthy subjects, together with their relative importance, what we call here the weighting. And the sensor column, what you see here, explains which of the sensors in the array is responsible for collecting the data described. The activity column here on the left is showing which test the feature has been collected. For instance, was it the get up and go from the chair? Was it while they were walking for 2 minutes? Was it while we asked them to stand and close their eyes and measure their sway? And the three columns here on the right are showing how much of the feature set is available depending on the extent of the sensor array used. And what you take away from all of this is that the lumbar sensor, the one that's placed just below your waist, provides the data for 10 out of the 17 features in this list, compares to just one feature that is dependent external sensor, so something that it's placed on your chest. And this prompts us to ask ourselves, what the results be were used alone? Of the relative rarity that you have with this PSP with any other disease you're studying with the Parkinson population. And I'll this part saying that the study was a diagnosis of PSP from the much commoner Parkinson's disease, and having a highly accurate, if you like, and specific diagnostic test is necessary to obtain a reliably PSP. It is also very critical running trials in Parkinson's to have patients with other disorders such as removed from the population as they tip. And of course, you have failed drug trials. And as you know, biomarkers to choose the right, stratify the right into trials. So some studies of wearable inertial have used only a single sensor, either just eye movements or just one sensor, you know, like your phone, or just a watch, if you like. Others have used, like we have been using, multiple sensors in these network arrays, as I've just shown you briefly in our latest work. And the key here is how complex an array is in performance. We found that the lumbar sensor yields the greatest amount of diagnostically useful information, but optimal performance, particularly with regard to specificity, required additional information from on the limbs. And what we concluded was that a wearable sensor array coupled with machine learning methods can accurately discriminate when you're looking into populations such as movement disorders, such as PD and PSP. And a choice of array complexity depends on context, really. For diagnostic purposes, a high specificity is needed, suggesting the more complete array is advantageous, so all 6 sensors, while for subsequent disease tracking, a simpler system may suffice. What we've been suggesting and what we're writing up some of our results now, where 3 sensors perhaps are useful. And of course, I'll finish off sort of on the research side of things on what we've been trying to do, looking at oculomotor now and coupling it with the gait in Parkinson's and the relationship between eye movements and freezing of gait during turning in individuals with Parkinson's. And just very briefly to say that Many individuals with Parkinson's experience a reduction in mobility, of course, with gait difficulties that include impaired turning that can trigger freezing. And these turning difficulties are particularly critical since falls during turning carry a higher risk, if you like, for someone to break their hip, have a hip fracture, compared to falls during a straight line walking. So when you're sort of walking along as opposed to having to turn to do something or go somewhere else. Also, it is well documented that Parkinson's patients are more than, I think, 3 or 4 times, if I remember correctly, more likely to sustain a hip fracture than an age-matched control without Parkinson's. And of course, these are public health costs that neither the UK NHS system nor the system in the USA and in Canada are able to handle at this stage. What we want to do here is really through use of kinematic and video oculographic analysis, along with perhaps some optokinetic simulation, to see whether individuals, for instance, who have turning difficulties show impairment in the ability to rapidly switch movement direction of their eyes and lower limbs, and whether the deficits that we have seen in their oculomotor function and sort of in the eye movement control contribute to these difficult Parkinson's, and whether we could potentially use these as a biomarker, if you like, in this disorder. I'm going to summarize here what I've tried to show you over the last little while. Eye movements are very useful in neurology and clinical neurosciences. Clinical eye movement examination can give us many clues, but it is a qualitative method. Technology can help us quantify eye movement abnormalities with great accuracy, saccades in particular, that are very well studied physiologically, both in macaques and in humans. We know what works, we know what doesn't work. We have very well, again, physiologically described the oculomotor pathway. We know exactly how its relationship is with the basal ganglia, so on. It's proven particularly valuable in movement disorders, and I see lots of colleagues from around the world contacting us, being very interested in how we interact with it, if you like. And my last point here of the fact that combining eye movement recordings with cognitive and gait technology can really provide a very powerful tool. And it's been really eye-opening for me, really, to be able to move from one modality to another, looking how to characterize our patient population in a way that helps us understand a little bit more physiologically what's happening and develop these biomarkers to capture this early signal that all of us are working towards, especially to help with the new clinical trials that we're bringing in. We do have an online population study on something slightly different, time perception in Parkinson's disease and age population. I'll send the slides over so anybody who wants to take part. This is an international study. It's run by my wonderful student, Lily Sue, who's in her last year of her PhD here. And you'll be asked to do sort of a visual task and then an auditory task, and we're looking at reaction. I won't tell you a lot, because if you do want to take part, I will bias you if I tell you what we're trying to do. Information here, so you can leave your email if you want to find out the results later. But if you are interested, we're interested in healthy controls, not only Parkinson's patients around the world. And I think the Parkinson's Canadian population, because Lily's Canadian, has been tremendous in helping us advertise this, and we've been getting hundreds of people sort of signing up. So please do participate if this is of interest to you. And because I know that there are various graduate students in the audience, I'll just say a little bit about academia, I guess, because I'm always asked about, what drive from bench to clinical and clinical to bench, and sort of try to make sense, if I could ever make sense, anything between the two. What I will suggest to the graduate students in the audience and anybody who might be looking at this later is find something that you're very passionate about. Find something that really interests you. It's a hard area to be in. You work on something that you really love, it makes it worthwhile. And of course, if you're passionate about it, when you get those negative results that are not really bad. We need to experiment and understand when things don't go well, especially I'm sure the colleagues know with electrophysiology and recordings and neurophysiology that things just don't go how you've planned them. Continue your hard work, and your passion and your interest will drive you. And of course, as I always say to my students here, it's a little bit of serendipity. You really need to be at the right time, at the right place with the right people who happen to take an interest in you for whatever reason. But again, within serendipity comes networking, which means that you need to go speak to people. And especially after COVID-19, you probably are almost used to doing Zoom, Teams. I don't know what you use in your institution, but try to present your work with your supervisor, find PIs and professors that can provide you advice, and try to do what you really think is worthwhile in terms of what you're interested in and your passion is. And again, more hard work and a bit more hard work, it always pays off. I'm going to finish off the talk this evening, well, this afternoon for you in Canada, by just giving you a couple of slides and leaving you with a little bit of inspiration, hopefully again to the graduate students there, of how you can combine your passions. So besides being a clinical neuroscientist, one of the passions I've always had is art, and how I can combine the two, giving voice to my patients. And I hope some in the audience might find this interesting. So one of the things that I've wanted to do is go outside the box, and instead of just focusing on the physiology and the neurophysiology and the biomarker work and the clinical work, is give voice to our patients so that we can understand what are we missing. That's fine if your eye movements are abnormal. It's fine if your gait is abnormal. But what else could I be looking into that could couple with my eye movements and my gait data that I haven't thought about, or I haven't had the time to let these patients explain to me. So one thing I've been leading for the medical sciences here at the University of Oxford is the art in neuroscience that Jeff very kindly said at the beginning. And this has been driven by, very frankly, I wanted to get out of the lab and of the clinics to go to the museum once in a while, because life in clinical neurosciences can be quite tough, especially when my eye movement work just was not working and it wasn't giving me the results that I wanted to at the beginning. Over the years, I've been awarded this fantastic opportunity by Vice Chancellor Awards to set up something called the Picturing Parkinson's, which is essentially the visions and voices of our patients, of the patients that myself and other colleagues see, who we don't understand, and we can't give anything. Even if I tell them your eye movements are abnormal, what does it mean to them? What does this disease do to them? Because they're all human at the end of the day. So what we've done over the last 2 years, I've had people contacting me from Canada, from US, wanting to do workshops online. So we had art workshops here in Oxford. And I thought, well, I can't just sit and wait for this pandemic to finish until I can do something with my patients. Let's do Zoom workshops and see if anybody attends. If we get 5 or 10 people, that's a success. We got 50 people who joined us from Canada and from the US. And in fact, one of the patients, Janice Grimes, who's also an accomplished musician in the USA, has written composed and written music for us that you will hear in a moment as part of her experience. So let me just show you a very brief clip that was put together by John Sargent, a wonderful artist who has Parkinson's, used to be a teacher, and has been working very closely with my team and I, and all these patients from all around the world, to put this exhibition together to amplify their voices. Because at the end of the day, you could do as much neurophysiology or physiology or clinics as you want. At the end of the day, is the patients that matter. So let me just show you this clip and I'll finish off in a second.
This work is the culmination of hours of thinking and talking and painting and writing. These are our narratives. We're people with Parkinson's. If you haven't got the time to listen to 3 minutes of this work, that's a shame, because this is powerful. This is the lived reality of living with Parkinson's. We shuffle into the offices of our neurologists and we ask to be understood and we ask to be listened to. And sometimes we come out feeling, I wish I'd said that. I wish I'd explained that better. Our art is our way of expressing our reality of living with Parkinson's. The discussions that come out in the groups, the work that comes out from that, the poetry, the images, these words, these are our realities. It's not sexy. Parkinson's isn't sexy. It's not quick. We aren't quick. We need time. We need time to move, to think, to do anything that we used to do without even thinking about. So that's why this film is the length it is. Every image you see, every word you read, even the music that's been composed and played is by somebody living with Parkinson's. These are our messages to you. If you want to understand what it's like to live with Parkinson's, whether it's as someone diagnosed or their carer, you need to listen to our voices. You need to understand the images that we're offering you. Say something about the way that we feel about living with this condition. The difficulties, the joys, the contradictions, the time element, the incessant pill taking. I've measured out my life in dosette boxes, not coffee spoons. Here are messages, they're powerful.
Okay, I'll leave it there. Right. It's been tremendous to be able to work with people with Parkinson's on all aspects, both of taking part in our research clinics, in our research with all sorts of testing I've shown you. And just to finish off, I have a tremendous team of people behind all of this. I don't do this alone. It's a collaborative work. I've got students, fellows, we're physiologists, neurophysiologists, clinicians, bioengineers. And I hope that, especially for the graduate students there, can see that hard work does pay off. It's not always positive results, but as I said, you learn from the non-positive results because you know what to drive next. And we do have fun while we do this, especially during Christmas. I think collaborative work is the way forward if we want to drive this area. Like, I think it's 8 o'clock and I'm right on time in finishing. Thank you very much for inviting me. And it's been a real pleasure to be able to share with you some of our work. I'm very happy to take any questions. Many thanks.